Hello everyone, my name is Ingrid Catarino. I am a PhD student in plant biology at UNESP, and I will present this work elevated to mitigate drought and increase the accumulation of 5-Caffeoylquinic acid and caffeine in Coffea arabica and mute drought stress. The concentration of carbon dioxide in the atmosphere is one of the main factors for the development of plants, and this concentration has been gradually increasing. Recently, the anthropogenic activities. The higher CO2 contribute to the increase of the greenhouse effect, which in turn cause climatic change, such as modification in precipitation patterns, favoring more frequent droughts. Water deficit is the main objective stress effect toward agricultural production. Climatic factors such as drought and high CO2 concentration are still poorly understood when related to plant metabolism. Therefore, this study aimed to investigate the effect of elevated CO2 concentration and progressive drought in the photosynthesis and specialized metabolites of Coffea arabica. For this work, coffee seed lines were grown in open-top chambers and subjected to high CO2 concentration or ambient CO2 concentration under well-watered and water-deficit conditions for 40 days. During the experimental period, measurement and harvesting of leaves were performed at the 0, 20, and 40 after water deficit position. In our results, we observed that the soil moisture was reduced at the 20 and 40 days, independently of CO2 concentration in comparison with the plant's water daily. The impact of the water deficit on the leaf's water potential was observed only at 40 days after the irrigation was stopped. The progressive water deficit reduced soil moisture, but the leaf's water potential decreased after 40 days of water withdrawal. We observed that in plants under high concentration of CO2, their rates of net carbon accumulation were always higher when compared to submitted plants for ambient CO2, even plants subjected to water deficits. The current concentration of atmospheric CO2 is a limiting factor for the photosynthesis of coffee trees, since they have a very low stomatal open and insensitivity to high levels of CO2. The maximum quantum efficient of photosystem to decrease only on the last day in plants submitted to CO2 elevated and water deficit compared to plants watered daily. This answer suggests that the coffee tree has an efficient capacity to dissipate excess energy in response to the imposed stress. The concentration of total soluble amino acids tend to increase under CO2 concentration, especially in plants with 40-day water deficit compared to trees subjected to concentration of ambient CO2 irrigated daily, while the content of phenolic compounds decreases under elevated CO2 concentration, regardless of water regimes. The amount of 5-Caffeoylquinic acid and caffeine increases in the plant and CO2 concentration and water deficit of the last day of the experiment when compared to the ambient CO2 plants. Taken together, these results suggest that elevating CO2 concentration mitigates mild water stress in Coffea arabica, increasing CO2 assimilation, instrumental efficient water use, and the content of 5-CQA and caffeine. A tendency to increase the caffeine contained in the treatment of high CO2 and water deficit indicates a positive effect of CO2 on the concentration of this compound in the leaves. Finally, we conclude that there was a positive effect of high CO2 concentration on photosynthetic rate and intrinsic water use efficiency, mitigating the possible negative effects of drugs and the influence of elevated CO2 concentration and drought on the accumulation of 5-CQA and caffeine throughout the experiment in leaves. I would like to thank FAPESP and CAPS, the Institute of Botany, and all those who collaborated with this work. Thank you for your attention.